Good evening. I'm Mr. Dotto, Principal of NFA North, and I'd like to welcome you to our annual Science Research Symposium. Before we begin, I would like to acknowledge that even under these unprecedented times and circumstances of virtual learning due to COVID-19, I'm really proud of all our faculty, staff, students, and families that have been supportive and have made it possible for our students to be here this evening to present in the symposium. I would also like to thank the members of the Scientific Review Committee who kindly accepted our invitation to support our students' research projects. We thank you. A special thank you to Ms. Oberly for her unwavering dedication to our students as they prepared their science research projects. Last but not least, I would like to thank the brilliant and hardworking students who researched, planned, and are now presenting these exceptional projects. Every year here at NFA North, the science research students select, conduct, analyze, and report their research. This year, more teachers were involved in supporting our students, both here on campus and virtually. Parents, your unwavering dedication to our science research program has made student success possible. Thank you for all your support. Now to our research students, specifically to our oral presenters. You are able to enjoy this wonderful experience due to your scientific curiosity and inquiry. I would like to challenge you to continue to push yourself by developing research skills, review literature, engage in discussions, analyze data, write, rewrite, and yes, let's not forget to have fun. Again, thank you for being part of this program this evening. Congratulations to our students. Have a great evening and enjoy the symposium.
Greetings and welcome to the 8th Annual Science Research Symposium at NFA North. My name is Kristen Oberly and I am the instructor of the Science Research Program. Even though this is our 8th symposium, it is our very first virtual event. Normally, the months before our symposium at school are spent planning for this event, both inside the classroom and after school, with students putting together the final touches at home. This year, the students have worked on everything independently at home, without being able to brainstorm with a teacher or a classmate beside them. The only time that we have had together as a class since April is twice a week on Google Meet, where students have practiced their presentation skills and fielded questions and comments from their classmates. Each one of these students has committed their time and energy to meeting at our designated class times, which on a Tuesday is 9:40 in the morning, every Tuesday since April. So at a time when most high school students would much rather be sleeping, our students have come to class ready to practice their presentation and gain valuable feedback from their classmates. That shows me how dedicated these students are to being part of our research team that we have solely created over the years. It is an honor and a privilege to work with such motivated young leaders who are committed not only to their own academic success, but also to the success of their classmates. We would like to thank Ms. Holmes for putting together this fabulous video for us, Ms. Sklarz for creating the symposium display on the district website, and to Mr. Dotto and Mr. Romano for helping me facilitate this virtual symposium into reality. Most of all, I would like to thank all of the science research students for their dedication to this challenging college level work that is part of this program, and to their parents who are there to support them through it all. And now the science research team invites you to enjoy the presentations that have been prepared for you. We also invite you to leave any questions, comments, or possible contacts and ideas that you have for students on the Google Forms that are attached to our website display. Thank you so much for spending your time with us today. We truly appreciate your support.
Morning, my name is Zachary John, and today I'm going to be presenting my research on microplastic concentrations in the Hudson River. So what are microplastics? Microplastics are pieces of plastic that are less than 5 millimeters in size. If you look to the photo on your right, you can see just how tiny microplastics really are. Microplastics occur from either the production of small pieces of plastic, including clothing fibers or small beads found in exfoliating skincare products, or from the breakdown of larger pieces of plastic. Microplastic pollution is one of the largest forms of pollution in the water today, and to this day, the effects of microplastics on humans is still not known. We do know, however, that the average person ingests 5 grams of microplastics a week, which is equivalent to 1 credit card, which is why there's an urgent need for more research. Review of literature. An article by Ritchie and Rosa in 2018 found that scientists have researched microplastic concentrations in order to best determine how to decrease pollution. An article by Mitch in 2019 described how, over the past few years, as a direct result of industrialization, plastic pollution has greatly increased. A study by Hudson River Park Estuary Lab in 2019 described how, in the Hudson River, microplastic pollution has increased the most of all types of pollution. An article by Parker in 2019 described methods to decrease plastic pollution. These include plastic bag and foam bans. In New York State, we enacted a plastic bag ban March 1st, 2020. However, due to COVID-19, the ban has been delayed. Lastly, a study by Hardy in 2019 found that studies have shown that microplastics are most likely in the drinking water in some areas. Specifically, this study stated that there was a high probability of microplastics being in the drinking water in some parts of New York City. Objective and hypothesis. The objective of my experiment is to quantify the microplastic concentrations in the Hudson River. I don't have a final research question yet, which is why I don't have a hypothesis. However, I do plan to look into how the size of the body of the water affects microplastic concentrations. Methodology. In order to quantify the microplastic concentrations, I first would have to collect multiple samples of water using a 0.3 millimeter mesh net in different locations. After that, I would filter the samples and let them completely dry. Then I would use a wet peroxide oxidation method, which would decay all the organic matter in the samples, leaving me with just the plastics. Then I would separate the microplastics based by size and then observe them under a microscope. Conclusion and expected results. Further research should be conducted on microplastics in order to determine the best way to decrease plastic pollution. The effects of ingesting a plastic is still not known. We do know, however, that in sea life, microplastics and plastic in general has been known to cause cancer and sickness, ultimately leading to death. Microplastics have been found nearly everywhere. They've been found at the top of Mount Everest and in the deepest oceans. Further research should be conducted on freshwater systems as large amounts of human activity occur in these waterways, such as fishing or swimming. In addition, these waterways are used for our drinking water, and most research has placed an emphasis on oceans compared to freshwater systems. I expect to find large amounts of microplastics in the Hudson River based off the previous studies and the previous readings that I have read. This is my work cited. My acknowledgement. I want to thank my classmates for always pointing me in the right direction whenever I had questions. Ms. Obley for giving me the resources I need to succeed in the class and always being there to motivate me. Mr. Pepperton for keeping me on track whenever I got distracted in class. Mr. Caldwell for giving me resources in the Hudson Valley that I could look to help further my research. And my parents for supporting my education journey. Thank you and have a great day.
Hello, my name is Deanna Regal, and my topic is the effect of the intestinal microbiota on oral drug metabolism. Intestinal microbiota, also known as gut flora or gut microbiota, are the microorganisms that live in the digestive tracts of humans and animals. The diversity of such microorganisms that live in the intestines assists with the degradation of nutrients prior to intestinal absorption. This is why intestinal bacteria composition is very important, for if there is an alteration in intestinal bacteria composition, it may lead to a different way the bacteria may break down oral drugs and food. Factors that alter intestinal bacteria composition include antibiotics, age, and diet. People, when they get an infection, take antibiotics, and some of them take probiotics to help replenish the intestinal bacteria composition that the antibiotics can destroy. In this study, we will find out how antibiotics affect your intestinal microbiota and describe the antibiotic and probiotic interaction with each other, the interaction with each other in their human intestines. According to Guinine and Cotter 2013, intestinal microbiota contains tens of trillions of microorganisms, including at least 1,000 different species of known bacteria with more than 3 million genes, which is 150 times more than human genes. They also said that the prevalence of varied bacteria species in the intestines have a commensal relationship, which aid in the breakdown of food into nutrients prior to intestinal absorption. Darcie and Judge 2017 states that everybody has a different enterotype or community of bacteria in their intestinal microbiota that likely results in varied nutrient or drug metabolism. The intestinal microbiota is such a crucial hidden organ that many common illnesses and diseases can occur if there is a maladaptation or imbalance in the intestinal flora. This is called dysbiosis. And dysbiosis has been shown to cause a variety of cardiovascular diseases, such as coronary artery disease and atherosclerosis, and other diseases such as obesity, gastritis, diabetes, and altered nutrient and drug metabolism. Also, according to Vads et al. 2018, drug metabolism due to intestinal dysbiosis will likely alter absorption and the intended mechanism of action for the drug. The objectives for this study include quantifying microbiota-mediated drug metabolism using liquid chromatography and mass spectrometry methods, screen representative drug classes to determine trends in microbiota-mediated drug metabolism, screen representative microbiota species to determine trends in microbiota-mediated drug metabolism, and screen probiotic commonly administered during antibiotic therapy to determine trends in probiotic-mediated drug metabolism. The hypotheses for this intended hypotheses for this study are that drugs are metabolized by the intestinal microbiota prior to intestinal absorption, and that probiotics metabolize the antibiotics prior to intestinal absorption. The intended methodology for this study includes in developing a microbiota representative bacterial library, selecting and analyzing a subset of GI microbial strains individually in heterogeneous colonies, selecting representative drug classes and analyzing a subset of drug structural motifs, and developing a liquid chromatography mass spectrometry method to quantify the parent drug and metabolites, and analyzing the concentration of antibiotics. This chart just shows the intended results based off the hypotheses. It is showing the concentration of a drug class of antibiotics called glycopeptides, in which the glycopeptides was placed in each of these three bacteria environments, such as Klebsiella, Bifidobacterium, and Staph bacteria. This shows the trend of the concentration of the concentration of antibiotics in each of these bacteria as time goes by. And as you can see, the concentration of these antibiotics in each of these bacteria decreases as time increases. This is just a continuation of the chart. This is just the chart placed into a line graph. As you can see, this is an indirect relationship that as the concentration of antibiotics in milligrams decreases, the time increases. And also as you can see, the three different type of bacteria metabolize the antibiotics at a different rate. In conclusion, this study will help create a better understanding of the intestinal microbiota and its role in antibiotic and probiotic metabolism, clarify how each of the enterotypes metabolize antibiotics and probiotics, improve antibiotic formulas and treatments, improve probiotic formulas, and improve treatments for people suffering with intestinal dysbiosis-related diseases. I would like to thank Major Milner for being my advisor and helping me with this presentation and additional information for this study, which I will be working with her with. And I would also like to thank Miss Overly, my peers, my friends, and parents for help motivating me, encouraging me, and helping me find additional information as well. Here's my works cited.
Good morning. My name is Amanda Waite, and my topic is how burn victims react to different treatment options. For vocabulary, I have 4 important words. Non-adherent means that the dressing will not stick to the wound and it allows for easy application and easy removal. Skin graft. Skin grafting is a surgical procedure that involves removing skin from one area of the body and moving it or transplanting it to a different area of the body. Collagen is the main structural protein in the extracellular matrix in the various consecutive tissues of the body. Botulinum, a liquid mixture of hydrocarbons that is present in certain rock strata and can be extracted. Severe skin burns are prevalent and a burdensome critical care problem. When working with these injuries, you focus on preventing infection while healing. There are currently 2 main types of treatments that are being used to treat burns. Skin Tempt, which is made of collagen, and Zeroform, which is made of petroleum. Here's an image of Skin Tempt and an image of Zeroform. For my review of literature, Zeroform is a non-adherent wound dressing. It is sterile and easily accessible in emergency situations. This was found in an article by Jenkins in 2018. Skin Tempt is a collagen-based wound dressing. It has many different layers to help add collagen proteins to the tissue. This was also found in the same article by Jenkins written in 2018. In an experiment done in 2017, Skin Tempt was proven to be more effective on skin grafts which are only used for the most severe of burns, which was found in an article by George in 2017. The best dressing is one that minimizes pain and adds proteins to skin, all while protecting it from infections. This was found in an article by Randall, published in 2020. The objective of my research is to find out which treatment option works best on burn patients that do not require skin grafts. My hypothesis is that Skin Tempt, which is the collagen-based skin graft, will prove to be more effective because it will protect the skin but also add proteins needed for the tissue to thrive and grow healthy and more abundantly. I intend to use a microscope to analyze and chart the properties of Skin Tempt and Zeroform. I also intend to analyze the normal skin cells, then treat them with the dressing. After which, I will analyze what the skin cells look like post-treatment and what the treatment itself looked like after the treatment has been completed. In conclusion, after conducting my research, I hope that patients or their proxies will have more information on the treatment options and how they work. This will help them make the best decision possible on what treatment they want to use. I would like to thank Dr. Maria David, my pediatrician, for being my mentor, Ms. Patricia Gould, my biology teacher, for helping me throughout my research, and Ms. Kristen Oberly, my science research teacher, for consulting me on my research. This is my works cited. Thank you very much, and that is the end of my presentation.
Salutations, ladies and gentlemen. My name is Sean Zamani, and this is my presentation on how PCBs affect fish populations in the Hudson River. Now, you may be asking yourself, what are PCBs? PCBs are polychlorinated biphenyls. They are a chemical compound that was used in the production of plastic before the 21st century. And my primary objective is to determine how PCBs are affecting different native fish species and if they are responsible for the mutations of these species. My review of literature. Now, there were 3 primary articles that I had used throughout my research this year. The first one was labeled Hudson River Rebounding After Decades of New York Sewer, which explains and sheds light on how the fish populations are increasing in size and schooling. The next article that I read was a National Geographic article labeled Hudson River Fish Evolve Toxic PCB Immunity, and this sheds light on the increased populations due to cleaner water, and it shows that the fish species are trying to develop an immunity to these chemicals. And the final article that I used was actually a research paper that was generously given to me by the DEC. And it showed how American shad, Atlantic sturgeon, and short-nosed sturgeon were affected by different chemicals. And the methodology that was used in this paper is an example of how I believe that I will do my research. My objective and hypothesis. My objective is to figure out how PCBs are affecting fish populations and species, and whether fish are developing to these toxicities. My hypothesis is that PCBs are negatively affecting the fish populations in the Hudson River, resulting in the death of many different species. My methodology. Now, it's very simple. I must obtain the samples of the different fish species, test for PCB toxicity levels, and evaluate in comparison to the results of uncontaminated or previously tested specimens of the same species. My anticipated results are that I hope to obtain that the specimens have a lower toxicity level than the comparative counterparts. And I hope to prove that there is such PCB immunities. In conclusion, I hope to shed light on the health and continued healing of the Hudson River, as well as its flora and fauna populations. The continued healing of the region will benefit all who live within and help to create a sustainable environment for generations to come. Now, these are my different works cited that I have used, and I would like to acknowledge Miss Amanda Higgs and Mr. Sean Madden from the DEC. They have been very generous in guiding me on my way to finding different information that I need. And I would also like to thank Miss Kristen Oberly, my science research teacher. Thank you.
Good afternoon. My name is Sommore Zola, and my topic is on the effect of exercise on the memory of early-stage dementia Alzheimer patients. Some people have been confused about what dementia and Alzheimer's is. Dementia is not a specific disease. However, it is a group of conditions that includes Alzheimer's disease as one of the most common conditions, characterized by the impairment of at least 2 brain functions, such as memory loss and judgment. Some people who have developed these diseases have had symptoms that may include forgetfulness, limited social skills, and thinking ability is so impaired that it interferes with daily functionings. Some studies have been conducted to show and prove that physical activities such as aerobic activities, which include running, walking, swimming, jogging, dancing, and other exercises, have actually improved memory and slowed down the process of developing memory loss diseases such as Alzheimer's and other memory loss conditions. One case study that has been reviewed is called Physical Activity in Dementia, the Need for Prevention. This case study conducted an experiment on over 5,000 women aged 55 or older. For this case study, only women were used due to the fact that Alzheimer's dementia affects women more than men. The only type of exercise that had been used in this case study was walking. Walking was used as their main focus exercise, which led to the results that walking in fact does delay dementia by at least 6 to 8 years. Another case study that had been reviewed is called Physical Exercise in Dementia. This case study experimented on people aged over 65. An exercise program was used to see whether exercise does in fact help people who suffer from these diseases and memory loss conditions. Some of the results showed that exercise not have only improved memory, but it also reduced stress and depression as well. and also showed that exercise can also help prevent muscle weakness, mobility problems, and other health complications with inactivity. Additionally, it can help promote a normal day-night routine, improve mood, and increase social participation. A third case study that has been reviewed is called Physical Exercise for Prevention of Dementia. This case study experimented on 180 people aged over 50. Those 180 people were broken down to three groups. The first group were people free of the disease. The second group were patients who suffer from early stages. The third group were patients who suffer from the late stages. The results show that exercise does in fact have improved memory and delayed the future possibility of developing memory loss conditions to those who don't suffer from these diseases yet. In my study, I will conduct a systematic review of studies demonstrating the effect of physical activity in improving memory in dementia and Alzheimer's patients. I'm planning at least gathering about 20 to 30 published studies on this topic and quantify them according to patient age range, gender differences, stage of diagnosis, treatment. I will also look at commonalities and differences in the type of physical activities used to be effective with patients. My main goal is to help determine specific treatments using physical activity that can improve memory and quality of life for those suffering from these diseases. Methodology. I'm planning on creating a chart and showing the commonalities and differences between these case studies. As you can see here, the first article is called Physical Exercise for Prevention of Dementia, and the age that have been used for this case study were people who were aged 50 and older, and the genders that were used were female and males. The types of physical activities used were treadmill, stationary bicycle, walking, jogging. The second case study is called Physical Exercise in Dementia. The age group that had been used was 5 to 12-year-olds, 12 to 18 years old, and adults and older adults 65 and over. The genders that have been used for this case study were females and males. The types of physical activities used were walking, dancing, jogging, bicycling, swimming, yoga, and Tai Chi. The third case study that has been reviewed is called Physical Activity in Dementia, the Need for Prevention. The age range that has been used for this is 55 and older. For this specific experiment, only females were used, and the types of physical activities used were walking. Those are the differences. Looking at the commonalities, they all prove that a physical activity does in fact improve memory and delays the possibility of having memory loss conditions in the future. Expected results. By conducting this systematic review, I am hoping to see that physical activity and exercise does in fact improve memory and delays the possibility for others to develop Alzheimer's, dementia, or any other memory loss conditions. Acknowledgements. I would like to thank Dr. Victoria Mano for helping me with this experience, with my project and experiment, and providing me with articles that I can read and review. I would also like to thank Dr. Claire for helping me with my systematic review and is still continuing to help me with my systematic review. Those are my citations that I've used for this presentation.
Hello, my name is Lila Amir, and for the past 2 years, I've been working towards identifying tears as a biomarker for ocular and systemic diseases. As a sophomore, I stumbled upon a magazine that claimed eyes are the windows to the body. At the time, I was grappling for a topic to base my research off of, and I took the whole eyes are a window to the body idea more literally than the next person. With additional research, I confirmed that the health of the eye is indicative of the health of the body. From there, I formed the claim that the complex composition of tear fluid presents as a future biomarker for systemic and ocular diseases. That's exactly what I set out to prove. This experiment aimed to expedite the healing process by introducing alternative methods to diagnosing diseases. From there, I hypothesized that by pinpointing compositional changes to tears using gel electrophoresis, pathways in disease can be identified. Each reviewed article greatly contributed to my research. As seen in the first box, the United States Department of Agriculture defines biomarkers as a measurable diagnostic indicator that is used to assess the risk or presence of disease. A manuscript detailing the extensive characterization of human tear fluid has been published by Marianne Dorr, titled The Investigation of the Global Tear Content from Healthy Human Tears. Moore writes, upcoming research in ophthalmology revolves around comparing healthy tears to diseased tears. Another researcher, Ms. Suzanne Hagen, wrote in her article titled Tear Fluid Biomarkers in Ocular Systemic Diseases, Potential Use for Protective, Preventative, and Personalized Medicine, that the application of tears are extensive, including systemic, neurological, and optical diseases. Mr. Evans Faulkner wrote in his 2001 article that lacroglobin is a possible biomarker for breast cancer. Lacroglobin is a protein secreted by the lacrimal glands that is closely associated with the cancerous tissue in patients with breast cancer. In addition to its breast cancer applications, lacroglobin was excessively present in patients with colon, prostate, lung, and ovarian cancer. By monitoring lacroglobin levels of tears in patients with a family history of cancer, the cancer can be caught early and treated before it progressed to an untreatable stage. As for my methodology, tears contain lipids, glycols, proteins, and metabolic wastes. Referred to as the omics, the study of each of these factors is the basis of this experiment. Tears from a patient suffering from a disease are extracted using the Schirmer strip method, which is inexpensive, non-invasive, and easily replicated. A mass spectrometer is used to ionize the collected chemicals and sort the ions based on their mass to charge ratio. Proteins, the main tear component that presents with abnormalities when diseased, is processed using a spectroscopic analytical procedure used to measure the concentration of proteins in a solution. The gel electrophoresis machine separates the macromolecules like DNA and RNA according to size and charge. The values of lipids, glycols, metabolic wastes, and proteins collected from each diseased patient are compared to the tears of a healthy individual. A difference in composition suggests an imbalance of homeostasis. An imbalance of homeostasis indicates illness. It is because the body systems are interconnected that scientists are able to relate abnormalities in the eye, such as glucose levels, to other unrelated systemic conditions like diabetes. And so, tears are a viable systemic and ocular biomarker. As to my expected results, depicted on the left is the varying amount of tear fluid collected from various parts of the feline eye. For example, there's less tear fluid on the cornea compared to the waterline. In my experiment, more tears means more information for me and less discomfort for the patient. On the right is a comparison of two people, one with Alzheimer's and one without. The individual with Alzheimer's, pictured on the right of each graph, consistently has a much higher protein concentration compared to the control, which is healthy tears. This offset in tear composition is indicative of a biomarker. To conclude, because ocular health is impacted by disparity in other regions of the body, tear fluid shifts in its hormones and chemicals in accordance to the imbalance of homeostasis. The elevated or depleted levels of these substances in the tear fluid can be identified using a mass spectrometer and gel electrophoresis machine. By comparing the abnormal tears of a diseased individual to the tear genome of a healthy individual, a correlation between systemic and ocular health of the chemicals in the tear film can be drawn. Routine tear exams, similar to that of blood work, will lead to earlier detection of diseases, which in turn calls for earlier prognosis and then earlier treatment, which improves quality of life and chances of survival. Here are my references, and for my acknowledgements, I would like to thank a few very important people, including my mentor, Dr. Kevin O'Donovan, for his wisdom, my science research instructors, Ms. Oberly and Mr. Pemberton, for their patience and support, as well as my science research peers for their constructive criticism. That brings my presentation on identifying tears as a biomarker for ocular systemic diseases to an end. Thank you for listening.
Is Darcie Guerra, and my topic is the effect of the type of bilingualism on the cognitive functions in children. For my review of literature, bilinguals have been proven to have better executive control. They have found that bilinguals have better attention and task switching capacities. Bilingual children perform better on stroke tests compared to monolinguals. bilingual advantages and executive control emerge at a young age. Many studies suggest that between second and fourth graders, children develop sufficient lexical knowledge to use orthographic structures. The strobe test is a neuropsychological test that measures executive function by inhibiting cognitive interference. Executive functions are a set of cognitive processes of selecting and successfully monitoring behaviors that facilitate the attainment of chosen goals. My problem is that studies on the benefits of bilingualism never focus on the different type of acquisition or fluency. My question is, does the type of bilingualism affect the proven benefits stated in previous studies? My hypothesis is, if a bilingual is more fluent in a second language and learned that at a young age, then they will have faster results compared to the other type of bilinguals. For my methodology, I'd send out surveys along with consent forms in a Google form with the strobe test. I'd have students take the exam 2 to 3 times and record their results. I'd then divide the groups based on the answers to the questionnaire into the type of bilingualisms. I'd then analyze the students' results. For the different type of bilingualism, the first is early bilingualism, and these are children who learn a second language before the age of 6. This is divided into 2 categories: simultaneous bilingualism, when 2 languages are learned at the same time since birth, or successive bilingualism, when a child has already acquired a first language, then learns a second language, which causes language development to be partly bilingual. The second is late bilingualism, which is learned after the age of 6. The acquisition of the first language is used as a guide to learn a second language. And the third is passive bilingualism, being able to understand a second language and not being able to speak it. For my human participants research, I'd have students from 2nd and 4th grades. The students and parents would fill out consent forms if allowed to participate, and the surveys would be administered online. The duration would be determined by participants, and I would have students without limitations such as being colorblind, visually impaired, having a learning disability, dyslexia, or extreme anxiety. For my informed consent process, I'd inform in my consent form about the study and how things would be performed. I'd explain that the experiment is voluntary and can be stopped at any time. The consent form would be detailed, explaining risks and benefits. Some risks can be about personal information or test anxiety. Solutions be to explain that the experiment is voluntary and anonymous. I'd inform that it isn't graded or isn't pass or fail. For my risk assessment, it would be minimized by being anonymous, and surveys would be confidential. For protection of privacy, surveys would be confidential. I would only ask questions about language acquisition and a little bit on background. I'd have no names, addresses, or numbers. For my data analysis, I'd have children divided by age and type of bilingualism. I'd record results of the strobe test, and I'd compare the results of the different types of bilingualism categories. These are examples of the questions I would ask. My goal is to see if there's a difference in the type of bilingualism, whether in fluency, age, or circumstance of acquisition, and to encourage others to learn a second language. My expectations are to have more fluent younger bilinguals with faster results on the strobe test. The benefits to the experiment are to increase knowledge on the benefits of bilingualism, to figure out if there is a difference in the ways the second languages are learned, or to motivate people to learn a second language at a young age. I'd like to thank my mentors, Dr. Jeffrey Rubin and Dr. Kathleen Guerra, my science research classmates, friends and family, and my teacher, Ms. Oberly. Thank you.
My name is Earnest Risa, and my research is on discovering, characterizing, and annotating a microbacterium foliorum phage, whom I named Vegas. To begin, bacteriophages are viruses that can infect and kill bacteria without any negative effect on human or animal cells. For this reason, it is suspected that they can be used alone or in combination with antibiotics to treat bacterial infections. In comparison to antibiotics, bacteriophages are supposed to have several other advantages. It is thought that bacteriophages are significantly safer and better tolerated, as they replicate only in the target bacterium but cannot infect mammalian cells. This conclusion seems supported by all the experiences gathered in the past in Eastern Europe and all the studies carried out more recently in experimental animals and humans, which have not reported significant adverse events following bacteriophage administration. There is also an estimation of 10 to the 31 phages on Earth, with each individual phage playing a vital role in their environment. Scientists are interested in phages because they help in understanding fundamental molecular biology such as vectors of gene transfer, drivers of bacterial evolution, and more recently as therapeutic agents. Moreover, the misuse of antibiotics has led to an increase in antibiotic-resistant bacteria, thus phages present as an alternative with their bacteria-killing ability. It is important to note phage therapy has not been approved in the United States, but ongoing research and clinical trials continue. Now, there are 3 problems explaining why research on phages should continue. Number 1 regards patchy knowledge. Little is known about phage diversity, whether phage genes truly represent a phage's abilities, and the interactions between host and phage. Second is the antibiotic resistance crisis. Bacteria are becoming increasingly resistant to antibiotics, but phages can be used as an alternative treatment. And third regards fundamentals of molecular biology. Phages helped with the identification of nucleotides and restriction enzymes. Plus, studying phages helps understand bacterial evolution. Moving forward, my objective is to exploit the phage I discovered in hopes that Vegas is novel, with genome annotation and bioinformatic analysis. This will help understand the mechanisms of Vegas's genes and provide future insight into the workings of bacteriophages. My hypothesis is, does Vegas identify as a novel Microbacterium foliorum phage? And if so, how much of Vegas's common and unique characteristics be characterized through bioinformatic analysis? Some important information to note is that my research has taken place in 2 parts. Part 1 had occurred in the summer of 2019 that involved isolation, purification, and amplification of Vegas. And Part 2, which will occur in the summer of 2020, involves genome annotation and bioinformatic analysis of Vegas. A simplified methodology of what I had done in the summer of 2019 started with enriched isolation. It involves amplifying the number of phages in the environmental sample that you would collect from the soil in order to get an exponentially larger concentration of phages specific to the host that I had chosen as Microbacterium foliorum. You would then perform a spot test, which involves screening for the presence of phages and waiting for visible plaques. The image to the right shows that phage plaques are clear zones that form when the phage has infected and killed the bacteria present on the plate. This shows that the phage indeed has the ability to infect and kill the host, and in my case, Vegas did infect and kill Microbacterium foliorum. You would then purify the phage, which involves obtaining a clonal phage population and ensuring that no contamination has occurred. And then you would amplify it to ensure that you have a high concentration of phage lysate to perform other experiments in the future. A simplified methodology of what I will do in the upcoming summer involves genome annotation, which is predicting gene functions, and then bioinformatic analysis. This involves using computer programs like DNA Master and BLAST or Primerator and any of the other programs available to help determine patterns in sequences and similarities to phage proteins. This can help answer questions like how similar are phages in a particular cluster, or what functions do the genes in the phage have. Predictive gene functions and proteins can provide substantial information about biology and the life cycle of the phage. In conclusion, going back to the hypothesis and objective of discovering Vegas's characteristics through genome annotation and bioinformatic analysis, this will aid in providing information on bacteriophages and their gene/protein functions. In turn, this will help give insights into the future of using bacteriophages as a treatment alternatives for bacterial infections, and nonetheless give insight into the workings of bacteriophages. As we find more and more phages and annotate their genomes, our knowledge of phage genomics increases, as does our ability to compare related phage expands. This is my works cited page to show my sources, and of course acknowledgments to Dr. Bala, my mentor over at the Mount St. Mary College, and Dr. Evan Merkhofer, who is also a professor at the Mount St. Mary College, who has helped guide me. Ms. Oberle, of course, my science research teacher, who has supported me through my research, and my fellow peers, friends, and family. Thank you for listening.
Are commercial cleaners used in hospitals as effective as they claim to be? Can herbal oils be a replacement for commercial cleaners? Good morning, my name is Stephanie Alvarado, and my topic is the effectiveness of herb-infused oils versus chemical cleaning products on bacteria reduction. According to the article, The Latest Dirt on Hospital Cleanliness, it states that hospitals are constantly cleaning using commercial cleaners, yet there's an increase in hospital-acquired infections due to multi-drug-resistant microorganisms, such as bacteria. Bacteria forms biofilms, which are groups of microorganisms that stick together to form an outer shell or barrier that protects the bacteria and allows it to survive. Biofilms are allowed to grow on both living and non-living surfaces, such as a sink or a doorknob. The presence of these biofilms are responsible for the bacteria's resistance to antibiotics and treatment.
leading to a focus on prevention strategies. The statement "kills 99% of germs" is one of the most used phrases that companies use to influence customers to buy their products. Once you hear the statement, you believe that all germs present would be clean, but that is false news. On the back of the Lysol, we see specific types of bacteria that these products can be used against, and it's not certain that it attacks these specific bacteria or even cleans it completely, based off my research. You might think maybe it's just a Lysol, but not at all. We see the same phrase more or less used on the 409 bottle. Once again, we see the types of bacteria in with the rest of the instructions. How about the Windex? We see the same phrase here, and it's interesting how the instructions are read through the bottle, which makes it harder for people to see. On the end of the page, in the smaller print, we see the types of bacteria the Windex works against. Lysol all-purpose cleaner has shown reduction of bacteria, but not the 99% the company states. Even more troubling, 409 multi-surface spray, which is normally used in hospitals, showed very little effect towards the bacteria. Interestingly, Windex glass cleaner spray showed the same amount of bacterial reduction as products not chemically equal, such as water. If chemical products such as 409, Windex, or Lysol are not the best options for killing bacteria, we must look to alternatives such as herb-infused oils. Aloe vera contains ingredients such as anthraquinone, phenolic compounds, prostaglandins, saponins, and glycoproteins. These compounds have an inhibitory action on both gram-negative and positive bacteria. Another herb, rosemary, showed phytochemicals such as camphor, caffeic acid, and antioxidants such as carnosous acid and carnosol. Due to these chemicals, rosemary is able to inhibit the growth of bacteria. The purpose of the study was to test herb-infused oils versus chemical cleaning products to compare which has a better outcome in reduction of bacteria on everyday surfaces. The study attempted to show whether herb-infused oils could be used as an alternate to chemical cleaning products at hospitals to reduce bacterial growth. It was expected that the bacterial plates that contain herb-infused oils would show a better bacterial reduction by 5% than the plates containing chemical cleaning products. It proven the hypothesis that natural-based cleaning products would reduce bacteria better than their synthetic chemical counterparts would assist in creating healthier environments for patients in hospitals and reduce the prevalence of resistant bacteria. For my methodology, I used these 3 cleaners based on the literature and these 2 herbs for comparison. As for my surfaces, I choose to use doorknobs, dump swab, the air, sinks, and phones. On day 1 of testing, it involved plating the bacteria and labeling. On the same day, I made different rosemary and aloe vera dilutions, which allows for different concentrations. To create these dilutions, 25.5 milliliters of water and 5.5 milliliters of ethyl alcohol were added to the oils. The only difference was that the ethyl alcohol was not added to the aloe vera due to it already being diluted once bought. Day 2 of my experiment consists of creating holes or wells in the agar and applying each product in their plate. Day 3 of testing was observation. Once we see that the microorganisms were growing, we decided that coming to the testing site on day 4 was unnecessary. Day 5 of the experiment was taking down the measurement of the zone of inhibition, which is the area around the well or the hole that shows bacterial reduction. Apart from that, we identified which microorganism formed, whether it was yeast, mold, or bacteria. While taking down the zone of inhibitions for my plates, it was shown that 409 contained 2 plates with the bacteria, both showing a type of reduction. Highlighted in yellow, 409 eliminated half an inch of the bacteria surrounding the well. On the bacterial plate containing a thumb, highlighted in green, 409 reduced 1/8 of an inch of the bacteria. Lysol on the phone, highlighted in blue, showed 3/8 of an inch reduced. The remainder of my results were either zeros or question marks. The zeros representing no elimination of bacteria, and the question marks mean there is no conclusive measurement. The empty spaces are places that had no bacterial growth, which was my study's focus. The hypothesis was that natural-based cleaners would be more effective by 5% compared to synthetic cleaners in reducing bacteria. Based on my chart, it showed that my hypothesis was rejected, due to commercial cleaning agents being more effective than herbal oils at reducing bacteria. My results showed that herbal cleaners had 0 effect against the bacterial reduction, regardless of the concentrations. I also received a lot of inconclusive results, demonstrated with the question marks. This could be due to factors such as time interval from each surface being cleaned. Would a sink that had been cleaned an hour ago versus 10 minutes ago have the same amount of bacteria? However, when further analyzing my results, it was shown that my hypothesis was neither supported or rejected. I was looking at the cleaners versus the control, which has shown that the herbal and chemical not only didn't eliminate bacteria, but enhanced the growth further. As we can see here, it seems as if the Windex and aloe vera had enhanced the growth of bacteria rather than reduced it. It is really difficult coming to a conclusion with my research due to the reason that I did not know the initial growth of the bacteria. Further research is needed to discover whether these cleaners enhanced the growth of bacteria or if an error was created when conducting my experiment. To conclude it all, my hypothesis was inconclusive. For future experiments, I would repeat the same procedure except lowering the amount of surfaces to at least 2, and obtain a plate with a known initial point of bacteria, or wait the 72 hours of growth. I'd like to thank Ms. Overly, my classmates, my designated supervisor, Mr. Ross Topliff, my mentor, Dr. Victoria Romano, Ms. Linda Romano, Ms. Patricia Gould, and Mr. Dean Keene. This is my works cited. Thank you for your time.
So, the American Society for the Prevention of Cruelty to Animals estimates that 5 to 7 million pets are admitted to shelters each year, with 60% of admitted dogs ultimately euthanized. Good morning, my name is Diana Ramos, and my science research topic is behavior traits detected in shelter dogs during interactions. Among the causes of relinquishment and euthanasia, behavior problems play an important role. The most common behavior reasons for returning dogs to shelters include aggression towards people, destructive behavior or vocalizing when left alone, excitability, and aggression towards other dogs. Thus, understanding the pattern of these problems is an essential key to establishing treatment and prevention programs. To improve the welfare of dogs, identify the need for behavior modification, and match dogs to new homes, shelter organizations first need to collect information about the dog's behavior. This information is used to find stable behavior tendencies to predict future behavior in similar situations. The human-animal relationship is defined as relatedness or distance between the animal and the human, the mutual perception which develops and expresses itself in the mutual behavior. A positive human-animal relationship is associated with pleasant emotions and rewarding events, whereas a negative relationship is characterized by unpleasant emotions and adverse-life events. Protopova 2011 found that an interaction session resulted in dogs having a decrease in heart rate, an increase in heart rate variability, and changes in behavior associated with a positive state of relaxation. Martinez 2019 found that attending puppy class may help prevent canine behavior problems as it reduces familiar dog aggression, non-social fear, and touch sensitivity. Dowell and Geyer 2011 found that results may enable a better understanding of a shelter dog's behavior, need for better behavior modification, and facilitate successful matches between dog and adopter because shelters can find stable behavior tendencies in individual shelter dogs and so make predictions of behavior in future situations. The present study aimed to observe the development of a shelter dog's understanding to specific commands as it took part in training sessions. The study attempted to display a positive correlation between a dog's understanding of the command and ability to complete the task as time passed. Based upon the results of earlier research, it was hypothesized that with the continuation of behavior modification programs already established in shelters, adoption rates would increase because of the dogs being taught to spend more time engaging in proper behavior and less time engaging in inappropriate behavior. Behavior modification programs are the systematic approach to changing behavior. Behavior modification looks to change a dog's reaction to a situation, a person, a thing, and an animal. These top behaviors are called on as tools in an overall program that helps to change how the dog thinks, feels, and acts. Establishment of behavior modification programs in animal shelters provides a variety of benefits, ranging from obedience training, allowing for predictable interactions, to environmental enhancement, increasing controllability of the environment. The main purpose of these programs is to improve adoption rates and reduce relinquishment rates of adopted dogs because of behavior problems. Five dogs residing at the Humane Society of Walden, a county animal shelter in Walden, New York, between September and October of 2019 were used for the study. Of this group, 4 of the dogs were male and 1 female. The sex ratio was also This sex ratio was also representative of the population of dogs at the shelter, where males outnumbered females. Age ranged approximately from 1 year to 9 years old, 40% of the sample size being 2 years old or younger. The dogs were of various and mixed breeds, and as is typical for most animal shelters, included strays and dogs abandoned or surrendered by their owners. Limited information was available on the history of each dog, so no attempt was made to distinguish them based on origin. So this slide is displaying the kennel cards for each of the dog participants. It lists their age, gender, breed, and then DOI, which is the date of intake. The Friday training sessions carried out in the Humane Society of Walden were devised to teach dogs how to behave and communicate effectively with people in everyday life. Classes took place for an hour during 4 consecutive weeks with the same dogs participating each week. The classes were conducted by animal behaviorist and trainer Anna Salanto, founder of Surulena Animal Rescue. In addition, some volunteer faculty members took part in the sessions under the supervision of the animal behaviorist referred to previously. All the sessions were run according to the exercise presented in this figure. So for the first exercise that they did was sitting, where the volunteers take 5 steps forwards and commands the dog to sit, instructed to maintain the treat in their hand below waist level as it allows the dog to acknowledge that he or she does not need to jump. The second exercise was touch. The volunteer directs their hand sideways and waits for the dog to touch their hand with the nose. Initially, they do not use the word touch until after 2 or 3 times. The dog to be able to understand the command first before having to hear the word. The third exercise was watch. The dog is firstly commanded into a sitting position, and then the volunteer brings the treat near their eyes and says watch. Once eye contact has been made, the treat is released right away. The fourth exercise is parallel walking. Volunteers are instructed to walk their dogs alongside other dogs to observe how they react and behave. This exercise also allows the shelter to pay attention as to how the dog walks on a leash. The primary aim of this study was to observe the development of a shelter dog's understanding of specific commands as he took part in training sessions. In correspondence to early research, it was expected for a positive correlation to be present between a dog's understanding of the command and the ability to complete the task as time passed. However, results from this experiment found it to be inconclusive to support the initial hypothesis. There was no effective way of analyzing the data collected, as only one of the 3 training sessions was recorded. In the following 2 classes, either the trainer was not present or the class took place in a different location. On the start day of the experiment, a video was recorded lasting 22 minutes and 35 seconds. 5 dogs were present taking part in exercises such as learning how to sit, touch, walk, watch, and parallel walk alongside other dogs. Taking into account 2 specific participants, Amos and Bella, there was observations to be mentioned. Upon the first week of meeting Bella, she was the most advanced, being able to understand the command. There was no knowing how many sessions she had attended, but in the week to follow, she did not take part in the class, having the opportunity of walking with potential adopters. Max had a very difficult time understanding a command. He would portray inappropriate behaviors such as panting, jumping on a volunteer, and pulling on a leash. But within the last session, he could sit when asked. One limitation of the study may have been that there was no attendance at the first of the 4 training sessions. The initial start of the experiment took place on September 20th, 2019, but the first training class had been a week prior. Another limitation presented in the study was the location. As stated, the first session took place in the parking lot of the shelter, whereas the following 2 classes were at a nearby barn. The barn occupied a greater amount of land. It allowed for volunteers to walk freely with the dogs to either the garden, the dock, playing fields, or to the playground. For future reference, a larger sample size and better recording of the training sessions would be recommended. This study failed to demonstrate that with the continuation of behavior modification programs already established in shelters, adoption rates would increase because of the dogs being taught to spend more time engaging in proper behavior and less time engaging in inappropriate behavior. Although 2 dogs in the study experienced change in behavior, it is not significant enough to support the original hypothesis. In view of limited resources and labor at most shelters, practical ways of implementing a training program have to be found. Observing human-dog relations will enable a better understanding of a shelter dog's behavior, need for behavioral modification, and facilitate successful matches between dog and adopter, because shelters are able to identify stable behavioral tendencies in individual shelter dogs and so make predictions of behavior in future situations. For my acknowledgement, all through the process of developing a research plan, I have received assistance from many individuals. Foremost, I would I would love to give sincere gratitude to my science research instructor, Ms. Oberly, and my classmates for guiding me through the research process and continuously providing feedback on my work. I would also love to give recognition to my mentor, Dr. Clara Thomas-Pino from the University of Maine, for always being there alongside me to assist and give advice throughout the troubles of my experiment. At last, I would greatly like to acknowledge the Humane Society of Walden for allowing me to observe and be part of the weekly training sessions. These are my purposes.
Hey guys, hope you enjoyed the video.
I know some parts of the video might not make sense, hence for this part of the video. But the main message of the video was friendship. Friendship in which is grown within 3 years. You get to know everyone in the program very closely, especially those who enter the year with you. I entered the science research class knowing no one, and in the end I got to meet many people who I consider friends. It doesn't matter if our classmates have graduated or not, because in a way it is as if we're still bonded together through our experiences in science research. I still have contact with our alumni, and it feels like no time has passed. It's the experiences that bond you together.
The environment that the classroom consists of is full of love and happiness. We laugh together at all the little mistakes. We provide feedback to what can be improved, and we cheer for those who struggle to get better. One of the main reasons this is the place that it is today is because of Ms. Oberly, who has allowed us to get to know each other and our interests, making it easy to motivate each other. Especially for me, who lacks in a lot of aspects, Ms. Oberly always found a way to guide me into the path I had to be on. I can speak for both me and Stephanie when I say that we are extremely grateful for the motivation and push you gave us, because we were great procrastinators and still are to this day, but we learn from our mistakes and try to do it earlier than later.
To our classmates and to the ones who are yet to come, you guys are the future. You guys have brought some bright ideas into consideration, whether it is regarding medicine or even environmentally. Follow what you believe is right for you, and choose a topic in which you sincerely are passionate about. Not only that, but build the program up, whether it is changing the color or font of an old program, or creating fundraisers for your experiment or for the overall classes. These are the memories that you keep with you and carry a long time. While I was screaming on top of my lungs at people in their cars to go to our car wash, I was laughing with the people I was doing it with, making it a memorable experience. Make the most out of what is happening, and in the end you will have things to laugh about.
All right, so that's it. Bye.
Bye.